The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.
Hello, welcome to episode 9 of the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. In the programme this month, how proteins chaperone each other, how bacteria work out which way they want to swim, and how new genes appear in the population. But first, to something that we spend a third of our lives doing, yet we understand virtually nothing about the process. Amita Sehgal is at the University of Pennsylvania.
So what we were trying to address is how sleep is regulated. The method we use is called a forward genetic screen. You mutagenize animals, and then you look for ones that are altered in your process or behavior of interest. In our case, that behavior would be sleep. And the system we use for these studies is the fruit fly, Drosophila melanogaster, because it really lends itself beautifully to this type of approach.
And it sounds like a rather daft question, but do flies sleep in the same way that we do?
So I don't know that I would go as far as to say that they sleep in the same way we do, but flies have a rest state that satisfies the criteria for sleep. And we have been using it to figure out how sleep is regulated. And a lot of what we're finding turns out to be also involved in controlling mammalian and human sleep.
So you found some flies that took less sleep. I presume otherwise this paper wouldn't exist. So what did you then do to try to find out why and how that was happening?
What we had used to generate these mutations is a chemical. It's called ethyl methensulfonate, EMS for short. And so what that does is it makes changes in the DNA. So what you then have to do is go and basically clone the gene that's responsible for the reduced sleep. And so, in other words, determine which of those many chemical changes in the DNA really accounts for the short sleep. And that is what we did.
So what genes came to fruition? What did you find when you did this study?
So at the end, we had it narrowed down to a region where there were 7 or 8 genes, and then we went through each of those to determine which was most likely. And for various reasons, we were able to exclude all the others. And then the one that we focused on was a change, a mutation in a nicotinic acetylcholine receptor.
Tell us a little bit about that receptor. Was it linked to sleep before? What do you think that the mutation is doing to it, and how did it affect the flies?
So the nicotinic receptor has been implicated in sleep before. Acetylcholine is thought to generally be wake-promoting, as is nicotine. So what was different about this one is that this turned out to be a receptor that does not promote wakefulness. It actually increases sleep. So when you mutate it, you have less sleep.
Does this mean if you increase the expression of that gene where you found this mutation, that this makes animals that need more sleep because it's a sleep-promoting gene?
Yeah, it's a great question, and the answer is no, with the caveat that you can never be absolutely certain that you have accomplished what you set out to do. So yes, we put this protein back in the fly. We tried to increase the expression to get more sleep, and we didn't get it, but it could be that our protein never really was increased in expression in, let's say, the right cell type or the right parts of the brain. And that could be because the animal naturally controls the levels of this.
So is it possible that in a normal fly, the expression of this gene is increased slowly over time as the animal becomes more relatively sleep deprived until it reaches a threshold point where it makes the animal go to sleep? Is that what you see? Because you would expect to see a sort of linear rise in the expression as an animal got sleepy if it were the trigger. So do you see that?
That is, in fact, what we see. We see that the levels of this protein are high at times of sleep. The only thing is that it does not seem to be a gradual rise. And we need to do more experiments to determine whether maybe it is a gradual rise, but our experiments so far haven't seen that yet because we haven't looked at close enough time points.
Do you think the alternative is then that something might be tripping it to turn on? Could it be just not there at very high levels to start with because something else, which is sensitive to how sleep deprived the animal is detecting the sleep deprivation level, and when it reaches a critical threshold, it then triggers your protein to turn on, and that's what triggers sleep?
That is the explanation we're currently favoring, yes. That it is not what we call the homeostatic signal, but maybe it is controlled by that other signal.
So what have you christened this protein, and how do you see it fitting into the genetic domino effect, for want of a better phrase, that we understand makes the circadian oscillator that's in the brain?
So we call this gene Redeye because that typically is associated with a sleep-deprived state. With respect to the circadian clock, we think that this is actually independent of the circadian clock. So there are two systems that drive sleep. There's the clock, the circadian clock, that makes sleep occur with the 24-hour rhythm. And then there is the other system that just ensures that you get enough sleep. And we think that this Redeye protein is part of that other system.
Amita Sehgal. Historically, a chaperone was someone who accompanied you when you went out, and they made sure, in the words of University of Michigan scientist James Bardwell, that inappropriate interactions didn't happen. Well, proteins also have chaperones, which are other proteins that help them to stay in shape and in the correct company. But how do they work?
Chaperones have been discovered back in the 60s, and it's been known for a long time that they help proteins get their proper shape. But the real mystery is how they could do that, just not for one protein, but for a whole bunch of proteins. Because, of course, every protein has a different shape, and how can you have one particular protein help a whole bunch of different shapes take place? There's been an idea for some time that these proteins that help other proteins fold have to be very flexible, because they would have to, of course, bind to proteins that have all different kinds of shapes to help them gain the proper shape. So we just started out by taking this chaperone that had the, I think, lovely name Spy, and we decided to try to make a better Spy.
So how did you seek to explore what Spy was doing and then adjust it?
Well, we had developed a way to force organisms to fold proteins better. We actually linked the proper folding of a protein to antibiotic resistance that an organism encoded. There's a gene that encodes resistance to ampicillin, so we could set things up so that an organism was in sort of a fold or die situation. They'd have to either improve their folding or they would be killed.
So how did you try to persuade the cells to optimize or improve their Spy?
Well, we just simply took Spy and we mutated it, and then we demanded a higher level of antibiotic resistance than it had before. Nearly all of the bugs died, but those few lucky ones that had improved Spy survived, and we looked to see what changes there were in those new Spies that were better than the Spy that nature had created.
So if you were able to improve on Spy, why hadn't nature done that already?
Oh, that's a brilliant question, actually. And we wondered that for quite a while. So what we did, actually, was we went back and we looked to see what nature had done to Spy in other organisms. The organism we worked in was E. coli. And interestingly, in other organisms, nature has, in many cases, come up with the same solutions we did. So then the question becomes, well, why isn't E. coli the best? And I think it boils down to the fact that evolution optimizes rather than maximizes. For instance, I work out sometimes with a trainer, and he's so muscle-bound that sometimes all his muscles just seize up and he can't even walk. So he's incredibly strong, but he's not very agile.
But on the other hand, if you can see how it's improved and why it's improved under certain circumstances, you can then begin to infer how it's doing what it's doing, how it's achieving that improvement.
Exactly, yeah. And that was the major motivation of our work, was to make something better, and then maybe we'd understand how it worked in the first place.
So how did you do that? Did you have to then look at crystals of Spy to see what the structure was in its wild type, the original form, and then the mutant, the better form, to see what had changed?
Yes, so we had a structure, so we could see at high resolution what the original spy looked like, and we knew what changes we had made. And the changes we'd made that made spy better were very, very specific.
And if you look at those tweaks, do they give you some insights into what the function of spy is in stabilizing those proteins, how it's doing its job normally?
Yes, so if you look at them, a good number of them, in fact the majority of them, work by making an area in the center of spy. Now spy has a sort of very thin cradle-shaped structure. It looks kind of like a cupped hand. A lot of the changes were right in the middle of the palm, and they made one part of the palm that we already knew was hydrophobic, in other words, sticky to oily substances. It made that part even oilier, even more hydrophobic. But it clearly wasn't just that, because it also seemed to make the protein much more flexible. So nearly all the mutations we got made the protein more able to wrap around its other proteins and sort of cradle them and protect them.
And now you've found this, what do you think are the key questions that you need to answer next?
Well, I think the real key question that we launched into in the paper was exactly what chaperones do to proteins. And I think that's really the key question. So far, we're starting to get glimpses of that. I mean, we meaning the whole chaperone community, and it's a long-standing problem about exactly what chaperones do to proteins to help them fold. And so I think that's really the key question that this work opens up, that at least for this particular chaperone, that we might be able to understand in, at atomic resolution what it looks like, a protein bound to a chaperone, for instance.
James Bardwell. You're listening to the eLife podcast with me, Chris Smith. Still to come, where new genes come from and how songbirds learn to sing their songs. But first, how bacteria control where they want to go. Patrick Mears.
So the point of this study, we were interested in how the number of flagella on an E. coli cell affects its behavior. So E. coli are these single-celled organisms, small bacteria, that have flagella that help them to swim. And the flagella basically look like corkscrews that rotate. And they can rotate either clockwise or counterclockwise, and they switch direction as the cell is swimming. And so the direction of rotation of the flagella affects how the cell moves. And the cell basically, when it swims, it has two modes. It has one that we call a run, where the cell tries to swim in a straight direction and go forward. And then occasionally it does what we call a tumble. And in the second tumbling mode, the cell pauses and sort of spins around and it eventually moves in a new direction.
So that must introduce something of a problem for the bacterium to have to overcome, because to produce meaningful net movement in one direction when it has variable numbers of these propulsion units, which appear to be acting independently in this way, how does it surmount that? How do the bacteria end up producing a purposeful movement in a given direction?
So generally, when the E. coli are swimming, all of the flagella rotate in a counterclockwise direction. And when they all rotate counterclockwise, they form a bundle where all of the flagella sort of wrap around each other and act as a propeller. And this pushes the cell forward. As I mentioned, the flagella can switch direction. And so occasionally one or maybe a few of the flagella will switch direction and rotate clockwise. And when this happens, it breaks this bundle. And that's when the cell pauses and changes direction. And so the question we were interested in was, how does the number of flagella actually affect this behavior? Because you might expect that a cell with more flagella would have a greater probability of having at least one of these flagella switch direction at any given time. And so our basic prediction was that cells with few flagella would only change direction once in a while, but not very often. Whereas a cell with many flagella, we expected to change direction much more often.
And is that what you saw?
No. To our surprise, what we saw was that a cell with one or two flagellum actually changed direction at about the same rate as a cell with 5, 6, or 7 flagella. So this was a big surprise to us, and it took us a while to figure out what was actually going on. To look at this, we used two different strains. So we used one strain in which the signal inside the cell that controls how frequently it's changing direction is controlled by the cell itself. And then we used a second mutant strain in which that signal was not controlled by the cell, but instead was fixed by a signal that we put into the cell.
And why should who has control make a difference? Why is that useful to you, being able to compare those two?
So in the strain where the cell controls it, these are the cells that are able to respond to the environment. And so when a cell is going in a direction that's good, it wants to continue going in that direction. And so it increases the probability of the flagella rotating counterclockwise. So basically, when the cell is going in a good direction, it tries to keep going in that direction. And when the cell realizes that it's going in a direction that's harmful or bad for the cell, it increases the probability of changing direction. And
I see. So this is quite neat. So basically the bacterium is influencing the probability of any flagellum changing its activity. And in this way, it is inducing a net change in direction, but it's not necessarily dictating which flagellum or cluster of flagella will do that.
Exactly. That's exactly correct. Then we looked at a second strain in which the signal is not controlled by the cell, but instead is fixed in time. And so the reason that we thought this might be affecting how cells with different number of flagella behave is because in the strain in which the cell controls the signal, at any given time, the signal might be very high or very low. And so if the signal is high, the probability of all of the flagella switching will be high. And so many of the flagella can switch potentially at the same time. On the other hand, in the strain where the signal does not change, the probability is constant, and so there's no compensation for the number of flagella. And basically what we found was that in the strain where the cell controls the signal, cells with one or two flagella behaved very similar to cells with many flagella. They changed direction at about the same rate.
So this is a rather natty and crafty, clever, in inverted commas, way for the bacteria to compensate for the fact they're continuously elaborating new flagella. And they may have many, they may have few, but this means the net result each time, regardless of number, is always going to be the same. It's going to be a purposeful swimming towards or away from an appropriate target.
Yeah, that's correct. So we often refer to this type of feature as being robust because cells, regardless of how many flagella they have, can behave similarly. And they've, like you said, they've developed this relatively clever system for compensating for that fact.
Patrick Mears, he's from the University of Illinois at Urbana-Champaign. Now, how do new or orphan genes crop up in a population, and where do they come from?
My name is Christian Flötterer. I work at the VetMed University in Vienna. I'm a population geneticist, and my interest is understanding adaptation and innovation that helps populations to adapt to a changing environment. That novel genes can arise at a high rate has been well documented in the literature. But if new genes arise at such a high rate, why doesn't the number of genes increase over time? Because that's not what you see.
And how do these orphan genes or new genes come about? What's their origin?
They arise from previously non-coding DNA. Basically, there is DNA there, and it is not coding for protein, and then suddenly it acquires a few mutations, and then it gets an open reading frame, and it suddenly has a function.
So at what rate are these orphan genes appearing? If you look in fruit flies, for example, in each generation, how many do you expect to see?
You don't see them in generations. You need hundreds of years.
So how did you actually approach this problem then, given that you've got that long time scale? They're appearing over hundreds of years to thousands of years. How did you do the study you've done?
Well, I should say maybe step one step back. How have been orphans detected beforehand? People were looking at basically isolated phylogenetic lineages, and they didn't look at closely related species. So what is novel in our study, that we looked at closely related species that were close enough that we could detect these novel genes there as well. And so what we did, we used a set of related species. We looked at relatives with an increasing divergence age. So they were at species that split from a common ancestor at different time points in the past. The key thing, we were not so interested in the origin of the new genes. We were interested in basically asking what is the persistence time? How long do they stay around?
In other words, by looking at an orphan gene that's cropped up but then subsequently disappeared, you can ask, well, how long do they persist in each given arm of the population or each individual species branch? And this gives you some insight into the persistence time of those orphan genes. And you can then ask whether some persist for longer or not.
Exactly. And what we found is that there are several indicators that give you a hint when genes are persisting for a longer time. One was the expression intensity. So if a gene is more highly expressed at a higher level, then this gene is more likely to be persisting. At the same time, we also find if a gene is more expressed in males than in females, this gene also has a higher chance to persist over longer evolutionary timescales than genes that have no bias.
Well, how do you account for that then, given that you have seen that? And if you think that it's a surprise that those genes should cling on, why do you think they do?
That is a good question. I wish I would have a good answer to this. One possibility could be that we can distinguish between spurious transcription and the true genuine transcription, because the way we identified whether a gene is basically a real gene is based on transcription only. And then we look at parts of the gene that are transcribed and have an open reading frame. This means they have a capacity to encode for a protein. And then these are basically classified as orphans. Nevertheless, it's well known that parts of the genome are just randomly transcribed. And so it could be that if you have a strong expression, this indicates that you really look at genuine signal rather than maybe spurious signal. Because I'm pretty sure what we have classified as orphans, there will be some noise in there that may not actually be real orphans.
What about the sex bias, though, as well? Because that's also very intriguing. Why should there be a favour on the male side to keep these things going?
Well, obviously you caught me on the wrong foot. I have no good explanation. There was an explanation why you could think that new genes when they originate are more male biased, because the idea was that you need simpler architecture of the regulatory module that basically is responsible for the expression pattern of a gene. But our data basically falsified this hypothesis. So we are left with an observation that's very difficult to understand.
Broadly, what do you think the implications are of what you've discovered? How would you, in a nutshell, say this is important because?
The importance of novel genes has for a long time been neglected. And so I think really to understand novelty in evolution, these new genes are probably one of the key factors that we need to understand better. And our study has made a major step in showing that these genes can persist for some time, but not all of them do. And these novel genes, if you apply population genetic tests, we see that there is selection preventing mutations occurring in these genes. And this is the best indication that there's something really functional.
Christian Schlotterer from the VetMed University of Vienna. And finally this month, how songbirds learn their songs. Duke University scientist Richard Mooney has been making recordings from neurons in the brains of finches in an area called the song premotor nucleus. These nerve cells appear to be crucial for the bird to produce and learn the song it sings. But, critically, the auditory input to these cells gets switched off when the bird actually sings itself, probably because the auditory signals would arrive after each part of the song has been sung.
A long-standing problem we're interested in solving is how the brain uses auditory information to guide vocal learning. In the region of the bird's brain we were recording from, neurons receive input from auditory parts of the brain, and in fact you can show that when the bird is quietly resting, those nerve cells will respond to complex auditory stimuli such as the bird's own song. But those same neurons are also active when the bird sings. And so the question was, are these neurons actually integrating auditory information as the bird sings, or are they flipping back and forth from an auditory function to a motor function as the bird goes from listening to singing? And the latter turned out to be the case, but we had to go a long way to find out that answer because it's a really tricky problem to answer in a singing bird.
Yes, indeed. So did you record and show that there was a sort of temporal relationship in terms of the activity in those sites to prove that?
Yeah, so in fact, the recordings involve recording from the same nerve cell in both states, and actually recording with a method that's really, really challenging to use even in a cultured neuron that's sitting in a dish, and that's something called intracellular recording, where we use a really, really fine glass micropipette. And when I say fine, the tip diameters are maybe a 50th of a micron. We can use these fine glass pipettes to poke inside of a small nerve cell, and we can record the little chatter of connections that are made onto that nerve cell from auditory and motor regions. So we can actually probe whether the auditory inputs are active when the bird was singing. And that's the real breakthrough in this study.
So in other words, is the bird listening to itself as it sings?
Well, we certainly know from behavioral experiments that it does listen to itself, but what we didn't know is whether or not these nerve cells, which seem so well suited to do that kind of self-monitoring, are actually listening as the bird sings. And they're not, even though we can show they get auditory input when the bird is quietly resting and listening to its song played through a speaker. Those nerve cells will get excited. But when the bird sings, the cell's active again, but the auditory input is switched off.
So what do you think the point is of having this feedback loop if most of the time it's suppressed when sound is coming out? Are the modifications based on audition to those cells made when there is not a motor program being executed? Is that how you think the system works?
So there's two parts to that answer. I mean, the first is that we think these nerve cells are doing something more than just controlling what the bird sings, but also help the bird identify and recognize the songs of other birds that have songs very, very similar to its own song. So that's part of the answer. The other kind of curious twist is that these nerve cells, their auditory properties, are clearly shaped by what the bird hears. Over development, they become tuned to what the bird sings. And other birds' songs that are very similar to the bird's own song will excite those nerve cells. Well, because the animal learns how to sing, that tuning property must reflect some kind of learning. So they're clearly responding to auditory experience, but when the bird is singing, the auditory input is turned off.
I read a study previously where scientists were looking at basketballers, and they showed members of the audience a ball leaving a thrower's hand, and some of those audience members were professional basketballers, others were coaches, others were just members of the public. The professional basketballers, they found, were effectively executing in their mind's eye the same throwing manoeuvre through their motor cortex that the pro was doing on the basketball court to make predictions, and were getting it right whether or not the ball would go in the basket about 90% of the time, compared with no better than chance for the coaches and members of the public who weren't doing that. Do you think you've got a similar situation here where these birds are effectively executing a sensory stimulus through a motor circuit to ask, would my song sound like this? And in this way, they're marrying up their song with what the other birds like them's song sounds like and doing a difference comparison?
I bet it is just like that. I mean, this is a very, very powerful idea. There's a so-called motor theory of speech perception that listening to speech sounds engages the motor circuitry in our brain that produces similar speech sounds, and that forms the basis of perception.
And if we were to put the whole thing together, can you sort of build us a model of actually how you see all this fitting together?
Right. So in the region that we're looking at, we think that this is a critical node for producing song, and you can demonstrate this switching this brain region off chemically with a local anesthetic. The bird will stop singing. So we know that it's essential for the bird to make song. What we guess, this song motor region produces a copy of the motor command and sends it into the auditory system. And that copy contains information about what should happen. If the two signals are highly congruent, then nothing happens. But if there's a mismatch, then the brain can say, I made a mistake, and can learn from it.
Richard Mooney. That's it for this month. Previous episodes of this podcast are available at elife.elifesciences.org/podcast, and all of the papers we've discussed are also there and freely available too. My name is Chris Smith. This is a Naked Scientists production, and do please join me again next time for another look inside eLife. Goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.